Myself, Dr.Viral Shukla, working as Assistant Professor in LG Institute of Applied Sciences. Dear students, today our aim of the practical is to study the ecological diversity of the bacteria. As we know that ecological diversity is the part of biological diversity. Now, ecological diversity is actually based on the bacterial requirement. Now, requirements are generally of two types. The first, that is nutritional requirement, and second one, that is a physiological requirement. Nutritional requirement is based on the requirement of carbohydrate as well as nitrogen sources, as well as mineral and vitamin growth factors, etc. And the physiological requirement, that is based on the pH or the temperature. Now, pH means what? See, each and every soil generally is having lot of buffering agents. So, some organisms are having the requirement of pH of below 7. So, that pH requirement is known as, or the bacteria who are growing below 7 pH or 6.5, they are known as acidophiles. Or if the organisms are able to grow below 4 pH, then they are known as extreme acidophiles. Some organisms who are able to grow between 5.5 to 6.5, they are known as acid tolerant. Some organisms who are able to grow at high temperature, for example 55 or above 55. So, the organisms who are able to grow above 55, they are known as thermophiles, hyperthermophiles. The organisms who are able to grow between 45 to 55, they are known as thermotolerant. And the organisms who are able to grow between 35 to 45, they are known as mesophiles. The organisms who are able to grow below 25 degrees centigrade, they are known as psychrophiles. So, on the basis of this kind of physiological requirement, the ecological diversity we can study. So, today, to study the ecological diversity, we are going to add a soil sample into the sterile 10 ml distilled water. You have to work between two burners. Mix it properly. Wait till the soil gets sedimented properly. Now, to study the ecological diversity, we have going to study the pH requirement and temperature requirement and salt requirement. As we discuss about the physiological requirement, the organisms are having requirement of salt. The organisms who are able to grow around at 30% of salt concentration, they are known as halophiles or halotolerant. These organisms always living in a hypersaline environment. So, this is 1 ml sterile pipette. Now, the soil is already sedimented. Take 0.1 ml of solution. Inoculate in a 5% NaCl concentration containing nutrient broth. Mix it well. Once again, take 0.1 solution. Inoculate into 10% salt concentration. Here salt is in a form of NaCl. And as we are using nutrient broth, see, nutrient broth is a generalized media for growth of aerobic organism, as it is containing 1% peptone, 0.5% NaCl and 0.3% yeast extract. Now the same soil suspension I am going to add into different pH to check the pH range of the organism which are surviving in a soil. So this is 2 pH solution, 0.1 ml suspension I am going to add. Mix it well. Once again 0.1 ml solution into pH 4. Then pH 6. During inoculation, you have to take care that your hands should be between the burners. Now pH 8. pH 9. And pH 10. So then mix it well, all the test tube. Put one negative control to check whether your medium is not contaminated or not. So after the completion of inoculation, we have to go with the incubation for 24 hours at 37° temperature in incubator. After completion of result, if you are getting the presence of growth between 4 to 6 pH, then that is known as acid tolerant organism. If you are getting the growth in a 5% or 10%, if you are getting the 10% growth, the growth of microorganism into the 10% salt concentration, then they are extreme halophiles. If you are getting the growth in a 5% salt concentration, then they are known as halotolerant. Thank you so much.